Hello everyone, I'm Kasuni. I'm a graduate student in Dr. Alan Pepper's lab at Texas A&M University. Today I'm going to talk about one of my projects in the lab that focuses on identifying serpentine adaptation genes by tracing evolutionary genomic history of Streptanthus, Caulanthus and their allied genera. One of the important questions in biology is that how plants adapt to extreme environments. Serpentine soil that I'm showing in this image is considered as an excellent model environment to do this, as this has high levels of heavy toxic metals like nickel, chromium, cobalt and low levels of essential plant nutrients like nitrogen, phosphorus, potassium. Serpentine soil also have low moisture holding capacities. These physical and chemical conditions make serpentine soil inhospitable for most of the plants, but there are some plants that are well adapted to serpentine soil. Among those plants we have Caulanthus amploxicolous variety barbarae, which we called as CAB. Interestingly, CAB sister taxon variety amploxicolous, which we called as CAA, is serpentine intolerant and grows on granite soil that has more balanced adequate nutrients. In this image, I am showing you the distribution of CAB and CAA. This can be found in California. CAA has a broader distribution compared to the limited distribution of CAB. In nature, these two plants do not overlap, but we can crossbreed them in laboratory conditions. Though the serpentine soil provides a stressful environment to CAB to live in nature, CAB is restricted to serpentine soil. This arises several questions like why CAB is able to live in serpentine soil, what kind of adaptations it has developed, and what kind of genes are involved in those adaptations. So these questions have led our lab to develop this two-plant system as a model to identify genes involved in serpentine tolerance. There are different approaches using in our lab to identify candidate genes for serpentine tolerance like coding sequence evolution, QTL mapping and RNA-seq. And in this study, I am exploring the use of gene ancestry as a novel possible tool to identify genes involved in serpentine tolerance. The evolutionary history of the serpentine tolerance of the streptanthoid complex, which CAB and CAA belong to, have been studied previously. And it looks like this, and this is published in Cacho et al. 2014. And in this phylogeny, the S represents Streptanthus and C represents Caulanthus. And we can see that in this phylogeny, they have put CAB and CAA together in this branch. And there is a large clade of Streptanthus taxa that are serpentine tolerant. And there are some individual lineages that have developed serpentine tolerance. This arises the question, did the serpentine tolerance in these lineages evolve independently, or were there have been some kind of gene transfer from other species, and if so, from which species? When building this phylogeny, they have not accounted for these complications like hybridization. So in my study, I would like to identify these relationships of this complex by taking possible hybridization into account. From previous studies in our lab, we have evidence that there have been two divergent events in the evolutionary history of CAB and CAA that looked like this. There have been a earlier divergence about 3 million years ago and a recent divergence about 0.25 million years ago that's supported by both nuclear and plastid data. And with this evidence, we have now this developed hypothesis that there have been an ancient streptanthoid ancestor that diverged 3 million years ago and come together, rehybridized and diverged, giving CAB and CAA. So does this hybridization play a role in serpentine tolerance evolutionary history? If this model is correct, we can hypothesize that the serpentine tolerant alleles in CAB has come from one of these parents. So if we have a region in CAB that have a functional relationship with serpentine tolerance, we can trace that portion of the genome in the evolutionary history to identify serpentine parent. And to trace parental lineages, we can use the other related Caulanthus and Streptanthus species. So when there have been hybridization in the evolutionary history, the best way to resolve that is to find the relationships using both organella and nuclear sequences. Often when there is an inconsistency in the nuclear and plastid phylogenies, this is an indication of hybridization. So that is why getting organella phylogenies are important in here. The aim of my study is to understand the evolutionary history of serpentine tolerance by building highly resolved phylogenies for streptanthoid complex using organella and nuclear gene sequences, and then utilize this knowledge of CAB and CAA gene ancestry as a new tool to identify serpentine tolerance candidate genes. This is the experimental outline of this project. I have used approximately 30 species related to CAB and CAA and has done sequencing for genomic fragments and also exon capture targeted fragments. And using those sequencing data, I have built 3 separate phylogenies for the chloroplast genome, mitochondrial genome, and nuclear genome. This is the chloroplast phylogeny that I built using maximum likelihood approach. And here I was able to resolve most of the clades with higher support. The blue color represents serpentine tolerance and red color represents serpentine intolerance. Here this is the CAB clade and this is the CAA clade. And in this phylogeny, the closest clade to CAB and CAA is a Caulanthus clade that has several serpentine intolerant taxa. In these plants, the organella genome represents their maternal lineage, so this suggests that the maternal lineage for CAB and CAA could be a serpentine intolerant Caulanthus lineage. This was supported by the mitochondrial phylogeny as well that showed a similar relationship to CAB and CAA with the Caulanthus clade. So with that evidence, we have this developed hypothesis now that the maternal lineage for CAB and CAA is a serpentine intolerant Caulanthus lineage. Here the circles represents their organella genomes and the dashed line represents the maternal lineage. So if the maternal lineage is the serpentine intolerant lineage, then the serpentine alleles in the CAB may have come from its paternal lineage, and could that be the Streptanthus lineage? So in order to identify the paternal lineage, we need to look at their nuclear data, which is a mosaic of both maternal and paternal genes. So I was able to build a nuclear phylogeny using single nucleotide polymorphisms in their nuclear genome, and I was able to resolve most of the clades with higher support. And when comparison to the chloroplast phylogeny, I saw that the nuclear phylogeny is discordant. Here the serpentine intolerant Caulanthus clade is distantly related to CAB and CAA, and the closest related clade to CAB and CAA is a Streptanthus clade that has several serpentine tolerant taxa. So this supports our hypothesis that the serpentine parent could be the paternal lineage, and that could be the Streptanthus lineage. But as I mentioned previously, nuclear data include both maternal and paternal genes. So in order to identify genes with paternal inheritance, we need to look at their phylogenetic history of each gene separately. So the next steps of this part of the project is looking at the evolutionary histories of nuclear genes with comparison to chloroplast tree. And here I'm hypothesizing that we would see one set of genes that follow an evolutionary history similar to chloroplast phylogeny, and there would be another set of genes that have a history different from the chloroplast phylogeny. So when working with the nuclear data, I'm going to take two pathways. One is I will pick a selected number of genes with higher coverage and make phylogenies for those genes to identify the ancestral lineage. And the second pathway is for the candidate genes that we can identify based on the other genomic data. We will sequence only those genes and look at their ancestry which we are hoping that would support our hypothesis more. So how we are going to use evidence from the evolutionary study to identify candidate genes? If we see an evidence of a clear ancestry for a serpentine parent for a gene that is already presented in the QTL region, this would be a good candidate gene for serpentine tolerance. Currently, I am in the process of making the gene phylogenes, and once we have those information from gene ancestry, I will incorporate that data with the other genomic data and prioritize candidate genes. Once we have a list of prioritized candidate genes, the next step is the functional validation of these candidate genes. And the outline for the functional validation project for my study looks like this, where I will transform the CAB allele for the serpentine tolerant candidate gene into wild-type Arabidopsis and its ortholog allele in CAA to wild-type Arabidopsis, and then wild-type Arabidopsis with an empty vector as a control. And I will also make Arabidopsis knockouts using CRISPR technology if necessary for the ortholog gene in Arabidopsis. And we'll grow these in different environmental conditions and do phenotyping comparisons to validate the function of the identified candidate genes. And I am currently in the process of this part of the project as well, and I'm doing this using few of the candidate genes that we have identified based on RNA-seq data. So finally, I would like to thank my PI, Dr. Alan Pepper, my committee members, and my lab mate, Elisa Grasa, for supporting with this study. Thank you very much for listening. Hope you all have a good day, and I'm happy to answer any questions you have.